Hello students and welcome to the lecture on allele frequency, genotype frequencies, Hardy-Weinberg law, role of natural selection, mutation, genetic drift, evolutionary genetics, genetic variation and speciation. The main objectives of today's lecture are to describe allele frequency and genotype frequency, to explain Hardy-Weinberg law, to give in brief the role of natural selection, to give the role of mutation, to explain genetic drift, to explain genetic variation and speciation. To begin with, let me first of all give a brief introduction about population genetics. Population genetics is the study of changes in the frequencies of alleles and genotypes within a population. Population genetics is a subfield of genetics that deals with genetic differences within and between populations and is a part of evolutionary biology. Population is a group of organisms of the same species usually found in clearly defined geographical area at a specific time. Now, let me begin with the first part of today's lecture, the allele frequency. Allele is one of the number of alternative forms of the same gene responsible for determining contrasting characteristics. The number of organisms in the population carrying a particular allele determines the allele frequency. For example, in humans, the frequency of the dominant allele for the production of pigment in the skin, hair and eyes is 0.99. The recessive allele, which is responsible for the lack of pigment, condition known as albinism, has a frequency of 0.01. This means that of the total number of alleles controlling production of the pigment, 1% result in a lack of pigment and 99% results in its presence. Since the total population represents 100% or 1, it can be seen that dominant allele frequency plus recessive allele frequency is equivalent to 1. Mathematically, frequency of A allele is equal to the number of A alleles by the total number of alleles, that is equivalent twice the number of capital A, that is the homozygote, plus number of capital A, small a, that is the heterozygote, by the twice the total number of individuals. Alternatively, equation P plus Q equal to 1 can be used to know allelic frequencies. After allele frequency, let us discuss the genotype frequency. The frequencies of particular alleles in the gene pool are of importance determining the frequency of genotypes. Since the genotype of an organism is the major factor determining its phenotype, calculations of genotype frequency are used in predicting possible outcomes of particular mating or crosses. It has great significance in horticulture, agriculture, and medicines. If an infinitely large random mating population is free from outside evolutionary forces, then the gene frequencies will not change over time and the frequencies in the next generation will be P square for the capital AA genotype, twice PQ for capital A small a genotype and Q square for the small AA genotype. The mathematical relationship between the frequencies of alleles and genotypes in the population was developed independently in 1908 by G.H. Hardy and W. Weinberg and is known as the Hardy-Weinberg equilibrium. The Hardy-Weinberg principle, also known as the Hardy-Weinberg equilibrium, states that alleles and genotype frequencies in a population will remain constant from generation to generation in the absence of other evolutionary influences. The frequency of dominant and recessive alleles in a population will remain constant from generation to generation provided certain conditions exist. Number 1, the population is large. Number 2, mating is random. Number 3, no mutations occur. Number 4, all genotypes are equally fertile. Number 5, generations do not overlap. Number 6, there is no immigration or migration from or into the population, that is, there is no gene flow between populations. Since one or more of these influences are typically present in real populations, the Hardy-Weinberg principle describes an ideal condition against which the effects of these influences can be analyzed. In the simplest case of a single locus with two alleles denoted by capital A and with the frequencies of capital A equal to P and of small a equal to Q respectively, the expected genotype frequencies are frequency for A equal to P square for the A homozygotes, frequency for small a equal to Q square for the small a homozygotes, and frequency A equal to twice PQ for the heterozygotes. The genotype proportions P square, twice PQ and Q square are called the Hardy-Weinberg proportions. The sum of all genotype frequencies of this case is the binomial expansion of the square of the sum of P and Q, and such a sum as it represents the total of all possibilities must be equal to 1. Therefore, P plus Q whole square is equal to P square plus twice PQ plus Q square, that is equal to 1. The realistic solution of this equation is Q is equal to 1 minus P. Since Hardy-Weinberg law describes an ideal condition, it makes use of certain assumptions that are: the allelic frequency at an autosomal locus in a population will not change from one generation to the next. The genotype frequencies of the population are determined in a predictable way by the allelic frequencies. If the allele frequencies in a population with two alleles at a locus are P and Q, then the expected genotype frequencies are P square, twice PQ and Q square. This frequency distribution will not change from generation to generation once a population is in Hardy-Weinberg equilibrium. The equilibrium is neutral, which means that a population deviated from its Hardy-Weinberg genotype frequencies will reach equilibrium after a single generation of random mating. But it will be new equilibrium if allele frequencies have changed. This property distinguishes a neutral equilibrium from a stable equilibrium, in which a perturbed system returns to the same equilibrium state. The main application is that the Hardy-Weinberg principle gives a mathematical baseline of a non-evolving population to compare evolving populations. If allele frequencies are recorded over a time and then expected frequencies are calculated based on Hardy-Weinberg values, the mechanism driving the population's evolution can be hypothesized. Our next subtopic is the role of natural selection. The concept of natural selection was put forward by Charles Darwin and Alfred Wallace in 1958. Natural selection is generally believed to be a prominent agent for determining the relative frequencies of alleles in a population and can be regarded as the machine that drives evolution. Natural selection differentiates between phenotypes in a population with respect to their ability to produce offspring. As genetic variants arise within a population, the fittest will be at the selective advantage and will be more likely to produce offspring than the rest. As the fit continue to enjoy greater survival, reproductively new species will eventually evolve. Some natural situations selectively allows one organism to develop and to propagate more efficiently, and one genotype is thereby afforded greater representation in the population's gene pool. If this selective process continues over many generations, allelic frequencies will change significantly and the potential will arise for evolutionary change. Natural selection process, in fact, influences the heritable portion of the phenotype. Natural selection includes the following parameters: survival rate, relative fitness, selection coefficient. If an individual has some form of a trait that gives him a better chance of survival in a given environment, evidently his offspring inherited the trait. This is known as differential survival. The adaptations are genetically controlled and heritable. Differential survival leads to differential reproduction of alleles responsible for the survival of the next generation. Natural selection means differential reproduction among members of the same gene pool. Natural selection can alter the frequency of heritable traits in three ways. These three ways of natural selection are: number 1, directional selection; number 2, stabilizing selection; number 3, disruptive selection. Let us discuss all the three of them one by one. Number 1, directional selection. In directional selection, one extreme of the trait distribution experiences selection against it. The result is that the population's trait distribution shifts towards the other extreme. In the case of such selection, the means of the population graph shifts. For example, evolution of the DDT-resistant insects and industrial melanism in peppered moth. Number 2, stabilizing selection. When selective pressures select against the two extremes of a trait, the population experiences stabilizing selection. Stabilizing selection keeps a population genetically constant. It favors the average or normal phenotypes and eliminates the extreme variants. For example, plant height might be acted on by stabilizing selection. A plant that is too short may not be able to compete with other plants for sunlight. However, extremely tall plants may be more susceptible to wind damage. Combined, these two selection pressures select to maintain plants of medium height. The number of plants of medium height will increase while the number of short and tall plants will decrease. Number 3, disruptive selection. In disruptive selection, selection pressures act against individuals in the middle of the trait distribution. The original population is disrupted into two more separate groups that later evolve into new species. If disruptive selection results in many new species, then it is termed as adaptive radiation. For example, the different beak sizes of the African fire-billed seed cracker finch, Pyronestus austrinus. Populations of these birds contain individuals with large and small beaks. Our next subtopic is the role of mutation. Mutation changes one form of a gene into another. Mutation is an evolutionary agent and mutability is a required property of the genetic material if evolution is to occur. Mutations are classified as beneficial, harmful, or neutral depending on whether they increase, decrease, or do not change the viability or fertility of their carriers. It is obvious that a mutation effect on fitness will influence its chance of persisting in a population. Many mutations are lost because they reduce the fitness of their carriers. Others are tolerated because they have been only negligible effects, and still others spread through the population because they improve fitness. Mutation rate cannot be too high, for it is the frequency of harmful mutations will be too greater for the population to survive. However, a mutation rate of 0 is also undesirable because mutation is an important source of genetic variation. Mutation may be highly directed at a particular locus such that allele A1 is selectively driven to the A2 form. Alternatively, the mutation process might be random, but with time, the A2 form would come to predominate over A1. Finally, mutation might provide a population with new alleles on which any and all evolutionary agents can act. After mutations, now we will discuss the role of genetic drift. Genetic drift is the random change in the allele frequency caused by sampling error across generations in a finite population. Small population size can exert a strong force on the evolution of populations through genetic drift. While it occurs in all populations, drift can be a major driving force for changing allele frequencies in small populations. If small populations are subjected to drift, the rate of evolutionary change can be speeded up dramatically. Allele and genotype frequencies can change unpredictably from one generation to next. The smaller the population, the more extreme these fluctuations tend to be. The actual change in allele frequency caused by genetic drift is random for any given generation. However, the effects of the drift will accumulate over time. Much of the mathematical theory describing the process of genetic drift was developed by Wright. Genetic drift has several important effects on evolution. Number 1, drift reduces the genetic variation in population. Number 2, it acts faster and has more drastic results in smaller populations. Number 3, it can contribute to speciation. Certain circumstances can result in genetic drift having a significant impact on a population. Two examples are the founder effect and the bottleneck effect. Now we will discuss the founder effect. When a small number of individuals known as founder population become isolated from a larger population, the smaller group may establish a new population whose gene pool differs from the source population. This is called the founder effect. This is essentially a sampling difference. The larger the subpopulation, the more likely it will resemble the parent population. For example, the evolution of picture-winged Drosophila species on Hawaiian island. Bottleneck effect. Sometimes the population will be reduced to a very small number. The genetic diversity of the small population may be much less than the previous population, leading to a great change in allele frequency. An evolutionary event in which the population size is radically reduced is known as genetic bottleneck. Genetic bottleneck increases genetic drift as the rate of drift is inversely proportional to the population size. For example, among houseflies in northern India, only a few representatives survive the winter months. These give rise to the summer population. The upcoming generation will represent the gene pool of those few flies that survived the winter. Now, let us discuss the second part of today's topic, that is evolutionary genetics. Evolutionary genetics is the study of how genetic variation leads to evolutionary change. It includes topics such as evolution of genome structure, the genetic basis of speciation and adaptation, and genetic change in response to selection within populations. Now we will discuss genetic variation and speciation. Genetic variation means that biological systems, individuals and populations are different over space. Each gene pool includes various alleles of gene. The variation occurs both within and among populations supported by individual carriers of the variant genes. Genetic variation is brought about fundamentally by random mutation, which is a permanent change in the chemical structure of the chromosome, and genetic recombination also produces changes within alleles. Mutations are sudden, unpredictable and inheritable changes which occur in the organism due to permanent changes in their chromosomes and genes. Mutations are of three types: genomic mutations, chromosomal aberrations, and gene mutations. Gene recombinations are new combinations of alleles of different genes which come together in an individual due to sexual reproduction. Because of them, no two individuals of a species are similar except for monozygotic twins. Gene recombination occurs due to dual parentage, independent assortment of chromosomes, crossing or random fusion of gametes and new alleles. Genetic variation among individuals within a population can be identified at a variety of levels. It's possible to identify genetic variation from observations of phenotypic variation in either quantitative traits that vary continuously and are coded for by many genes, example, leg length in dogs, or discrete traits, those traits that fall into discrete categories and are coded by one or a few genes, example, white, pink, red petal color in certain flowers. Genetic variation can also be identified by examining variation at the level of enzymes using the process of protein electrophoresis. Ultimately, genetic variation is caused by variation in the order of bases in the nucleotides in genes. New technology now allows scientists to directly sequence DNA. Examination of DNA has shown genetic variation in both coding and in the non-coding intron region of genes. Genetic variation will result in phenotypic variation if variation in the order of nucleotides in the DNA sequence results in a difference in the order of amino acids in proteins coded by that DNA sequence, and if the resultant difference in amino acid sequence influences the shape and thus the function of the enzyme. For a given genome of a multicellular organism, genetic variation may be acquired in somatic cells or inherited through the germline. Genetic variation can be divided into different forms according to the size and type of genomic variation. Small-scale sequence variation, less than 1 clue base pairs, include base pair substitution and indels. Large-scale structural variation, more than 1 clue base pair, can be either copy number variation, that is loss or gain, or chromosomes rearrangement like translocation, inversion, or segmental acquired uniparental disomy. Numerical variation in the whole chromosomes or genomes can either be polyploidy or aneuploidy. Now, moving to the last part of today's lecture, that is the speciation. Speciation is the evolutionary process by which biological populations evolve to become distinct species. The biologist Orator Cook coined the term speciation in 1906. Charles Darwin was the first to describe the role of natural selection in speciation in his book, The Origin of Species. There are 4 geographic modes of speciation in nature based on the extent to which speciating populations are isolated from one another. Number 1 allopatric, number 2 peripatric, number 3 parapatric, and number 4 sympatric. Speciation may also be induced artificially through animal husbandry, agriculture, or laboratory experiments. Now let us discuss allopatric speciation. During allopatric speciation, a population splits into two geographically isolated populations. For example, by habitat fragmentation due to geographical change such as mountain formation. The isolated populations then undergo genotypic or phenotypic divergence as they become subjected to dissimilar selective pressures. They independently undergo genetic drift. Different mutations arise in the two populations. When the populations come back into contact, they have evolved such that they are reproductively isolated and are no longer capable of exchanging genetic material. For example, Xiphophorus variatus found in the south is divided into 3 distinct populations. Each population has its own ecological preference and is found in 3 distinct rivers. All these species, Xiphophorus variatus, Xiphophorus videum, and Xiphophorus ebeliani, have different morphological features by which their localities can be identified. Now, peripatric speciation. In peripatric speciation, a subform of allopatric speciation, new species are formed in isolated smaller peripheral populations that are prevented from exchanging genes within the main population. It's related to the concept of founder effect since small populations often undergo bottlenecks. Genetic drift is often proposed to play a significant role in peripatric speciation. Now moving on to parapatric speciation. In parapatric speciation, there is only partial separation of the zones of two diverging populations afforded by geography. Individuals of each species may come in contact or cross habitats from time to time, but reduced fitness of the heterozygote leads to the selection for behaviors or mechanisms that prevent their interbreeding. In parapatric speciation, there is no specific extrinsic barrier to the gene flow. The population is continuous, but nevertheless it does not mate randomly. Habitat differences may be more important in the development of reproductive isolation than the isolation time. For example, ring species like Larus gulls. Now, sympatric speciation. Sympatric species are two or more related but reproductively isolated populations. These occur in close proximity in the same or overlapping geographical areas. They occupy the same microhabitat or locality, share the breeding conditions but do not interbreed or do not produce fertile hybrid. Sympatric speciation refers to the formation of two or more descendant species from a single ancestral species, all occupying the same geographic location. 2 basic points about sympatric speciation are: number 1, speciation due to difference in ecological niche of same population. Number 2, unlike the allopatric speciation, in sympatric speciation, the intrinsic factors play a very crucial role. The intrinsic factors may be genetic factors, which include the whole genetic makeup of the population. When these factors are in action, after a considerable time, a population may re-establish in races, and this is the beginning of the speciation. The best illustrated example of sympatric speciation is that of the cichlids of East Africa inhabiting the Rift Valley lakes, particularly Lake Victoria and Lake Malawi. There are over 800 described species, and according to estimate, there could be well over 1600 species in the region. All the species have diversified from a common ancestral fish, the Japanese rice fish Oryzis latipis, about 113 million years ago. Their evolution is cited as an example of both natural and sexual selection. Sympatric speciation driven by ecological factor may also account for the extraordinary diversity of crustaceans living in the depths of Siberia's Lake Baikal. Students, that's all about today's program. Hope you have understood and enjoyed. Thank you and goodbye.